Hello, my name is Ryan. I am a laboratory assistant here with the Cognition, Action, and Neurodynamics Lab at Smith-Kettlewell. One particular interest of our lab is the use of haptics and how we particularly perceive haptics. So, for example, Braille is a highly well-known haptics that is used by blind and low-vision individuals to perceive text. They sweep their fingers across specific patterns, specialized patterns of dots, and do that, they transform that into meaningful text. It pretty much serves as the replacement as if us sighted individuals were reading text or listening to text, but for blind and low vision individuals, that is sweeping their finger across these braille patterns, and that is their equivalent of text. For this particular experiment, what we're trying to look at is how does the brain respond to braille? How does the brain perceive braille? And we're combining it with this method called EEG, where we are measuring the brain signal while one is reading braille. And there we hope to find how does the brain respond to braille. We may also see at what timeline does the brain process braille. So we have an idea of does it take seconds, is it millisecond, is it an hour? How long does it take for the brain to from one sweeping their finger over the braille text to actually knowing in their brain, oh, okay, this is the letter A or this is a vowel. This experiment will help us kind of determine that timing, but also how does the brain respond to braille. In addition to Braille, one thing that's interesting to note is that Braille interacted with different neurotypes. So, for example, ADHD, dyslexia, that is a common disorder that may affect text processing in regular sighted individuals. But for blind and low vision individuals, we have no idea exactly how does that interact with Braille. And so this particular experiment helps us get one step closer to there. In order to understand how we may come up with strategies to address these pitfalls of ADHD and dyslexia in Braille, we first must need to understand how Braille alone, just normal blind and low vision individuals, how do they perceive Braille? How does the brain work with Braille? Once we know those, then can we take those findings and apply it to potentially ADHD and dyslexia and other types of neurodiversity?